Good morning, everybody. All right, time to rock and roll. So, what we're going to be doing today is starting a kind of a new section, kind of in a way carrying on a little bit of what we did with DNA replication uh, last section. Let me share my screen. Well, you don't need to see that. Where are we at? Here we go. All right, so essentially we are starting right here. And what we're going to do is give you a brief kind of look at, eukaryotic genome structure and organization and kind of the things that make our genome so large, particularly compared with bacteria, um, and how actually that's it's kind of very moving target, to be honest. And then we're going to spend, after we get through that and talk about transposons, which are particularly neat things, we're going to spend the rest of the time really on what's called the central dogma, which is actually wrong. It should be the sequence hypothesis. It's really gene expression, right? So how we go from DNA through an mRNA intermediate. Sorry. To getting a functional product, which is typically, not always, protein. Ooh, I'm allergic to all of the very many animals. I've just been playing with puppies, so because they're digging a hole in their bed. It's one of the reasons why I'm sneezing. Anyway, and so as you can see by the amount of time that's being spent on transcription and translation, they are very involved, complex things to study. And so this section will be very much, it's going to be a lot more abstract than a lot of the math-based stuff that we've worked with so far. And it's going to rely a lot on remembering what different things do, right? So there's an awful lot of details involved in this because those details are very important. We can give you kind of a general Cliff Notes view of the overall principles, but without those details, those principles are largely meaningless, right? And so, and this is going to be a very important kind of prelude to the bulk of what we're going to cover in the last section, which is on gene regulation, right? And that is a huge subject, one which I've in my life only really barely scratched at. In fact, it's funny when I started my postdoc at Penn State, there was some journal clubs, right, which is basically where you every week someone picks a paper, reads it, prepares a presentation on it, and everybody else kind of gets to read it and have it explained to them, right? That's kind of what a journal club is. And there was one on gene regulation called the Gene Reg Journal Club. And I was like, oh, that sounds interesting. And one of the PhD students was like, oh, no, you don't want to join that. No, don't do that. Because it's just crazy, the level of detail that you can go into, right? It's a real rabbit hole to drop down into. And so what we're going to be learning about transcription and translation in this section is going to be really important to get a lot of the complexity of the control of gene expression in the next section, particularly eukaryotic gene regulation, which is, as one would expect, given our increased complexity, far, far more involved and subtle with many more players than bacterial gene regulation. Sorry. Okay, so we're going to start today by just giving a general overview of, get back up here, kind of how eukaryotic genomes are structured and some of the challenges involved in that, some of the oddities involved with that too. And also kind of giving you just a general overview of like packaging as well, which is quite a big deal in eukaryotes, because obviously prokaryotes, packaging of DNA is less of an issue for two reasons. One, they have much, much smaller genomes than eukaryotes do. And two, they don't have to package it into a nucleus because they don't have one. It's kind of concentrated in like one part of the cell, generally speaking, but they don't have the packaging issues that eukaryotes do. Okay, so this is what's called a karyotype. This is basically what your chromosomes would look like when they are fully condensed, right? So they don't look like this normally, and they're not pretty colors either. These are, I'm not entirely sure what has been added here. I don't know if these are different, you know, markers, maybe immunostaining, perhaps with antibodies or something else. I don't actually know even where this picture came from. But this is to kind of give you a sense of what chromosomes look like relative to each other, right? So you're already familiar with the whole idea of X's and Y's being very different in size, right? And so Y is very much a truncated chromosome, but it's not kind of out in the woods in terms of how small it is, because, you know, our other chromosomes, particularly 21 and 22, are approximately similar in size, right? And they're ordered in size. Basically, this was how, you know, they were first characterized, and they just gave them one as biggest, you know, 22 as smallest. 21, as you can see, this is the chromosome that, if you have 3 copies of it, leads to Down syndrome. And so this is also a kind of a hint as to why you can actually even get trisomy of chromosome 21. It's very small. There aren't a huge number of genes present there. Actually, if you look at it, there are 351 genes on chromosome 21, right? So the likelihood of having an extra one of those 351 genes screwing things up, developmentally speaking, is smaller, not zero, smaller than it would be, say, for chromosome 1, where you have over 3,000 genes, right? And so trisomy is very rare, right? Actually, Down syndrome, however, marked the changes are in terms of development, in terms of cognition, you know, visible differences even, right? Personality as well. That's a pretty mild version of trisomy. If you have more than 2 of most of these other ones, you don't make it. Essentially, it results in a miscarriage if even it gets that far. Okay, so anyway, so you can see there's a lot of variation here, right? And when we get to gene density, we'll see that that's even more the case. However, one of the really kind of curious things is what's called the C-value enigma or C-value paradox, right? And the C-value is basically the haploid genome size, right? So we have 23 chromosomes. And that's our N value, right? That's the number of unique chromosomes that we have. Obviously, you know, we have 46, right? Because we're diploid. But the actual amount of unique information we have is 23 chromosomes, right? And the C value is basically how big, right? If you kind of stretch all those out and count how many bases they have, right? How big, hello, girl. My dog coming downstairs. She's a teenager. How big that all of those bases added together is your C value, essentially, right? And so the human genome, obviously, we have twice that much DNA because we're diploid, but our actual genome is about 3 billion bases in size, or 3 billion base pairs, right? And as you can see, we're kind of middle of the pack, to be honest, right? In fact, we have, I don't even know what all these different protists are. I'm not much of a taxonomist, right? But we're at the upper end of whatever euglenozoa are. I don't know. Small things because they're protists, right? Unicellular organisms, right? So in terms of our genome size, we're not particularly remarkable, right? In fact, compared gymnosperms, oh, type of plant, I can't remember. Angiosperms like trees, I think. Anyway, again, not much of a taxonomist. We're way at the bottom range of that. So even though we consider ourselves complex organisms, and many of us consider ourselves the peak of evolution, in terms of genome size, we're pretty, you know, moderate. I mean, I guess we could feel superior about ourselves relative to sponges. Like, hey, we're way more complex than sponges are. It's like, yeah, congratulations. We're more complex than sponges. Sponges they're not even bilaterally symmetrical. They're just, well, they're sponges, right? Kind of useful if you want to have a shower, but they're sponges. Anyway, so this is called the C value enigma or C value paradox, right? How can we, as complex macroorganisms, right, have a genome size which is kind of middle of the road if we're a earthworm, right? Or which are annelids. And I'm a little bit better with multicellular organisms or animals. Similar for mollusks, about middle of the road for crustaceans, right? So I don't know many crustaceans personally. I don't even know many crustaceans in general, right? So I don't even know if we have a bigger or smaller genome than a lobster, for example, or a crawfish, or crawdad if you're from certain parts of the US, right? So what's that all about, essentially? And it's really crazy. It kind of gets really extreme, right? I'll come back to that in a little bit. So these are just some units, right? So kilobase KB, megabase MB, gigabase GB. These are just give you units, right? So if say, you know, organism size is 400 MB, that's 400 million bases, basically, megabases. This is tachyfugu, which is otherwise known as pufferfish. Also immortalized by an episode of The Simpsons too. I don't know if anybody's ever watched that. Stuck in my head. Basically, Homer wants to learn how to cut tachyfugu up so that he doesn't get poisoned and he doesn't succeed. Anyway, and this thing, whatever this is, I have absolutely no idea. It looks like a snake, but also looks like a skink that's lost its legs. Has a far, far larger genome than this thing does. Yeah, this one swims, this one slithers. You know, there's no real kind of explanation as why this has, or at least from face value, why it has a much larger genome or C value, right? And it actually can get really extreme. I don't know where, actually I should have looked this up. Does anybody know what has one of the largest genomes in terms of C value, the number of base pairs? What organism has, I think actually is the largest genome. Some of you may have had some of it today for breakfast. Anybody got a guess? This is always fun to ask. Find out what people eat for breakfast too. Oh, cool, Raelio. I'm glad someone else has seen that episode of Simpsons. I'm kind of pretty outdated when it comes to my Simpsons. Not chickens? Nope. Eggs? Nope. That actually comes from chickens. Pigs? Nope. I'll give you lettuce? No. Yes. Jelly, not cows. Wheat. Wheat has a genome which is 17 gigabases. Right. So the wheat genome is, what's that? Oh, I'm not very good at math. That's over 6 times the size of our genome. So those tacos or tortillas or whatever you had for breakfast, I had mini wheats, something like that. So the genome of that organism, right, that was used to create those things that we ate is 6 times larger than a human genome. And yet we're the one eating the wheat, right? We're the ones that actually figured out how to process wheat so we could make tortillas, right? So clearly there's no linear relationship between organismal complexity and size of our genome. In fact, the wheat genome is the size that it is in part because it's the result of 2 hybridization events, right? So it actually has the equivalent of three semi-independent genomes, right? And so the original ancestor to wheat basically hybridized with another plant and then again hybridized a second time to create what we, consider wheat. It's actually very similar to, oh shoot, what's the name of the ancestor of maize? Let me just double check. Come on. You see at the side of the road around here, which I'm, yeah, teosinte, that's it. Got a memory like a sieve, right? So if you've seen this stuff, what does it look like? Here we go. Growing at the side of the road, which you will do in particularly southern Texas and obviously in Mexico as well. This is the ancestor of modern maize or corn, right? And it also arose through a series of genome duplications, right? So whole genomes just getting doubled through some mechanism that I don't fully understand, right? And so you actually get a lot of these funky events in the history of particularly domesticated plants. I'm not so sure about animals, but plants certainly seem to be kind of more prone to these hybridization and duplication events than animals do, right? And so that's in large part why we have this huge range over here. So, and actually even not just the C value, like the actual content of the haploid genome, but even the number of genes which we encode in our genome isn't hugely variable, right? So, shoot, actually, oh, this is kind of embarrassing. I should kind of gen up on my taxonomy before this class. I'm trying to think where nematodes are in this. They're probably in annelids somewhere, maybe. Not even sure that they're on there, right? But they're kind of like down on the list of complexity, I guess. But anyway, nematodes, what I work on, right? C. elegans. It has approximately the same number of genes as a human does. give or take, about 20,000. And yet it has 969 cells. They're actually few enough that you can count them if you so choose to do so, right? And it has a much smaller genome than humans do too, yet it has the same number of genes and is less complex as an organism in some ways, right? Other ways, it's far more complex than we are, depending on your perspective, right? So there's very little relationship between genome size, gene number, and organismal complexity. In fact, the only real correlation that I've been able to, kind of like fumble around and understand is that as an organism gets more complex, or the more complex an organism is, the more chromosomes it's likely to have. That's about it. That's the only, and again, that doesn't hold true particularly well for things that have had duplication events like wheat, for example, because obviously they're going to have more genomes, more chromosomes rather. So that's about it. So and this whole C-value enigma or paradox and how we are complex organisms, right? How we have complex behaviors and structures and so on and so forth, right? That really, truly isn't best explained by what we're going to study in the last quarter of the course, which is the control of gene expression, right? And that's why I kind of spend a bunch of time talking about it now, because like this kind of thing, because it's really key to understanding like the complexity of us as organisms and the complexity of our genomes. And so you're probably wondering, okay, so if we're about here, right, and we have 3 billion base pairs, C. elegans, I can't even remember how many base pairs it has. I think 100 million, something like that. Same number of genes, what makes up the rest, right? And essentially, most of our genome is made up of other stuff. So about, I can't remember the exact number, to be honest. I should look it up sometime. 3 to 5% of our genome is taken up by protein coding genes. So those 20,000 genes are basically encoded by that much of our genome. Little bit. And so what's the rest? Well, a lot of the rest is what's called repetitive sequences and satellite DNA, right? So these are, and also other things like transposons, what's commonly referred to as junk DNA, right? Actually, there's an awful lot of interesting stuff that goes on with junk DNA. So this is kind of like an outdated view. But basically, it's a lot of repetitive DNA, a lot of transposons, which we'll cover on Wednesday, and short tandem repeats, right? And so these repeats are sometimes as short as 3 base pairs, right? Really short repeats that are repeated over and over and over again. These may be the result of DNA polymerase stuttering, right? So just slipping on those repeats. Sometimes they're the result of movement of transposons around in our genome. There are lots of different reasons for it, right? And these are actually, in many ways, these satellite DNA is how we can genotype or type individuals. And actually, that's in large part what our kind of our fingerprint is, our DNA fingerprint. Whenever you see like CSI stuff, they're not actually sequencing our genome. They're just looking at variation in what bits of satellite DNA we have, right? Because those are obviously inherited, same as everything else. And so in many ways, this kind of these repetitive sequences of satellite DNA and transposons and just kind of evolutionary cruft, basically, just kind of evolutionary baggage genetically, are what explain a large part of the difference in the C value of an organism, right? It's not the protein coding stuff, like the core requirement, it's everything else. And so there's a lot of different types of kind of repeats and transposons and junk and just various assorted stuff. And these are range from single copy sequences. So these may be genes, right, which are in the haploid genome at least present once. Transposons, other bits of DNA which are there only once, maybe because I haven't had time to be copied or whatever. We've got a smaller amount of kind of things which are present tens to hundreds of times. And we have an absolute load of stuff which is present thousands of times over and over and over and over again. And actually, one of the things we've already covered that are fall under this category are telomeres, right? So those buffers on the ends of our chromosomes that protect against chromosome shortening due to the 5' primer problem, right, consist of these highly repetitive sequences, right? That's actually how they can be extended by telomerase, right? So this is one way in which we've actually benefited from, sorry, watching my dog on the couch wagging when my wife arrives. These are actually hugely beneficial to us because obviously they protect our protein coding regions from chromosome shortening. Here they are. Okay. Hey, love, Luna hasn't been out yet. She came down when I was teaching. All right. And so if you look at the individual chromosomes that we have in our genome, you can obviously see that there's a huge variation even amongst chromosomes as well. And so we range from, what's the lowest density? Can't remember off the top of my head. Probably the Y chromosome, because that kind of wins most of the prizes when it comes to being small and not very useful. But that has a gene density of 6 genes per million bases, right? Each gene varies in size quite considerably from, I don't know, it can be from, a few thousand base pairs to tens of thousands of base pairs, right? But gene density is very, very low on the Y chromosome. And you can get as high as, 15, I think is that the highest? No, even 19 genes, 27 genes per megabase, right? So chromosome 19, it's only 72 million bases long, has 1,700 genes, right? So that's more than chromosome 4, right, in terms of genes, which is, what's that? Probably 2.5, nearly 3 times the size, right? And so there's no real correlation between chromosome size and number of genes either. Obviously, the smaller they get, the less genes that they can actually fit on there, but that's not really the kind of main determinant. Okay, so aside from this, we have the issue of fitting DNA into the nucleus, right? And the issue with that is that well, there's a couple of kind of compounding issues there. One is that our eukaryotic genomes are huge, right? Just in physical size, right? If you just, stretched out, I think the old saw is that if you took every cell in your body and stretched out the DNA from end to end, it will reach the moon or something like that, right? So in terms of like physical size, they're actually pretty huge. And yet our nucleus, or the nuclei in our cells, are fairly small, right? And not only is, so, I mean, I don't know if you've ever done a quick pack to go on holiday or something, you can stuff a lot of things in a small space real quickly. But if you actually want to get at them, it's a different matter. So that's the conundrum, is that A, we have to fit our genome into a very small space, but yet at the same time, we have to be able to access it, either to copy it for DNA replication for cell division, or to transcribe it as part of gene expression, right? To be able to turn that information into something functional. And so there's a couple of ways around that. One is supercoiling, right? So if you remember what we talked about in DNA replication about DNA gyrase and topoisomerase relieving supercoiling, right, ahead of the replication fork, well, you can actually introduce it as well to be able to basically coil the DNA up into a denser structure. And so the main workhorse in terms of fitting DNA into the nucleus. Oh, hang on, got a question. Don't know why chat doesn't pop up sometimes. No, not necessarily, because so the Y chromosome contains relatively few genes, and most of them are involved in sex determination, right? So there's a little bit involved in testes development, the SRY region, right? The sex controlling region Y. There's some that are involved in development of male-specific organs and features, things like that. But the vast majority of what makes you you is encoded by genes on your autosomes. So neither the X nor the Y contributes a huge amount to kind of what you can actually see. Mostly it's from the autosomes. And so you get that equally from your mother and your father. So yeah, it's actually very hard sometimes when you kind of think about genetics in terms of physical appearances, because A, you're only looking at a small subset of what makes you, right? It's kind of what is expressed visibly on the outside. And B, there's an awful lot of pattern matching, right? So as humans, like hunter-gatherers, we're very good at finding patterns, right? Oh, that looks like a dog, it's a dog, right? Kind of thing. And so when you're actually looking for patterns like that, you can actually see things which don't have as much weight as maybe they do in reality. But because of the way you see stuff, it's a lot more likely that you're going to associate those things. So yeah, it's kind of pretty much 50-50 from both parents. Okay, so in terms of, good question though, but in terms of compaction, the large part of how we compact our DNA to fit into our nucleus and still be able to access it to be able to express those genes is through the formation of chromatin, right? And varying the types of chromatin as well, which we'll get to later, because there's two different types of chromatin. We've got, come on, heterochromatin, right, which is still made up of the same components, right, which are basically, come on, histones and DNA, right? So both heterochromatin and euchromatin are both made up of, they're the same thing, but it varies in terms of degree of coiling and accessibility. So that chromatin can vary between heterochromatin, right, which is condensed, kind of inaccessible. So the correct term is transcriptionally repressed, right? So what genes are there tend not to be expressed, right? doesn't actually have very many genes present there. It kind of makes sense, right? You don't really want many genes there. And those regions tend to be around the centromeres and at the telomeres. So the centromere isn't actually a region of the chromosome that has like a little round circle in it. It's just a region which is typically heterochromatin. Doesn't even have to be in the middle, could be at the end as well, right? So you always see it like this. It's not always that simple, essentially. Telomeres are heterochromatin kind of makes sense because really their purpose is as, pardon me, buffers during DNA replication. Not really entirely sure why the area around the centromere is heterochromatin too, to be honest. I know that that's recognized by other proteins which form the kinetochore, which is really the bit that spindle fibers attached to in mitosis and meiosis. But why that forms there, I don't know. Partly perhaps because of how many, like the percentage of satellite DNA, maybe. I don't actually really know, to be honest. And then between those two extremes, between the telomeres and between the transcription repressed DNA at the center of the chromosome or at the centromere, you have euchromatin, right? And euchromatin is chromatin that is much more accessible or can be more accessible. This is also can be dynamic, right? You can have regions of the chromosome which are shut down, heterochromatin, which in a different context, time of development or whatever, are actually open and accessible. They're euchromatin, right? So this isn't just difference by region of the chromosome, right? Ends, middle versus bits in between. This is also something which can change for the same part of the genome, depending on context. And so this is in many ways, and we'll come back to this when we look at control of gene expression, this is a very higher order way of controlling gene expression. So this is only shut down, the euchromatin is only shut down during cell division. And we'll get to why that is later. And most of the genes are found on the arms of the chromosome. If you remember back to when we were talking about linkage, right, how map distance was not the same as physical distance. Again, that kind of comes back to that issue as well. Okay, kind of bouncing around a wee bit, just bring up the chat again, make sure I don't miss any questions. So essentially chromatin is DNA and protein, and the main proteins are histones, right? These are proteins which are highly conserved, but you actually use a whole bunch of them. It's not just one single protein. And when you wrap DNA around histones, you get something called a nucleosome, right? And this is essentially a bit of DNA wrapped around a complex of histones, right? And that looks like this, the whole beads on a string deal, right? And so essentially you have, I think it's 2 and a bit wraps around the protein, and you have a stretch of DNA called linker DNA in between the two. This linker DNA actually has several purposes. One, it allows these things to twist and get wrapped tighter, potentially. And two, the other kind of main one, is you can actually slide these beads up and down the DNA, right? So these are not static in place things. They're things that can vary in location. And typically there's about 200 base pairs of DNA between the start of one nucleosome and the start of the next one. And that consists of the DNA that's wrapped around the core particle, right? About 145, 147 base pairs, and the bit of DNA that links those particles together, that linker DNA. Right, so it's about every 200 base pairs, you will find a nucleosome, give or take. Okay, and so these nucleosomes actually consist of, well, really 9 proteins, right? We've got 4 pairs of proteins. These are the histone molecules that make up that kind of that ball, right? These are actually encoded by different genes, right? And these are also things that can be modified. So if you remember me talking about DNA methylation in the context of mismatch repair, well, histones can also be methylated and acetylated to change their properties, right? Which we'll get to when we talk about gene expression, or the control of, at least. So we've got 8 histones in the core particle, right? And these have tails which kind of stick out the side, like kind of frilly bits. And we have a 9th protein called the H1 protein, which kind of glues everything together. And so if you look at this kind of like zoom out, right? So we're kind of super zoomed in here, right, to these individual kind of beads on the string. If you zoom out a bit, you can get higher order chromatin structures, right? And this is typically what most DNA looks like in the cell, right? During interphase, right? Or when cells are not dividing, right? So it doesn't look like this during cell division, or at least in the M phase part of cell division, because that's it changes quite a lot there. It basically goes down to this kind of compaction, right? So in most kind of transcriptionally active cells, I guess would be the best way of putting it, your DNA is going to look something like this, right? It's going to be a long, long fiber, maybe kind of looped up, organized in particular regions, which we'll get to in a second. If it's being heavily transcribed, it may kind of stretch out a little bit, maybe. Can also be transcribed like this. If it's going to be shut down in terms of heterochromatin, might be bound a little tighter, but you can also, heterochromatin also means other things in terms of accessibility as well. And then when you start entering prophase, essentially that kind of chromatin structure gets packed down even further into what is now, you can see these kind of X-shaped chromosomes, right? So you will not see these X-shaped chromosomes at any time other than basically prophase and metaphase, or prophase, prometaphase, and metaphase. Right? They do not exist at any other time. Most of the time, your DNA is actually kind of packaged in really funky ways, actually. This is kind of, it's something that's really been better understood probably over the last 10 years, maybe, as more basically cooler. I wonder if I can find a picture of that. Let's see if I can bring up some cool pictures. Oh, there you go. So, oh, what's my dog doing? You can actually label different regions of different chromosomes. It's going to move this out of the way. And if this is a nucleus of a cell, don't even know what, nah, don't know what cell that is. You can actually see where different chromosomes localize, right? And what they tend to do is they actually get anchored to the inside of the nuclear membrane, right? So you have a nucleus like so, it's got lots of holes in it. And each chromosome is actually kind of stuck to the periphery, right? The inside of that nuclear membrane. And you can actually visualize where they are. Like you can, this is with in situ hybridization. So this is basically using probes that recognize particular sequences that have a fluorophore attached to them that glows a different color. And so if you use ones for different chromosomes, you can actually go, well, that's chromosome 12, this is 14, this is 15, right? So they're physically stuck to that nuclear membrane, right? And even better, I don't know if you can, come on, get that later. I'm not entirely sure what they're showing here, but maybe this is what I'm thinking of. It's kind of hard. The search terms are difficult to use, but you can actually tell when chromosomes are being accessed as well, right? And you can see that this is a dynamic process. So parts of, actually, I think that's what this is showing. This is probably the nucleolus. You can actually see when different parts of different chromosomes are expressed, right? When they're open as a euchromatin versus heterochromatin. So this is a hugely dynamic process. And if you were to kind of look at it as a kind of map, a color coding map, you'll see that it looks kind of like this, where each individual chromosome kind of responding to different colors has its own different kind of territory within the nucleus, which is just crazy, right? Like the degree of how well we understand stuff now is just getting nuts. Like the kinds of things that we can do as scientists just continually amazes me. It really does. And you can actually look at how this changes in a living cell, right? Actually, even if you can get a thin enough organism, in an actual living organism as well, not just tissue culture, which is just, I just think it's amazing. I was, hang on, let's see if I can. Discovered this the other day. So this is a complete tangent. This is what a mitochondria looks like. This is the crista, you know, the folds. I didn't realize that ATP synthase actually doesn't work alone. It works as a dimer, either as a V or a U shape. And that is actually what shapes the inner membrane of mitochondria, right? This is with, what do they call it? Cryotopomography or something like that. Just absolutely astounding. Really is. Anyway. And so to be able to divide up these chromosomes, right? So if this is a mitosis, then to separate the sister chromatids, or if it's meiosis, to separate the chromosomes themselves. These chromosomes have to be condensed even further down to these entirely inactive condensed chromosomes, right? So when you see chromosomes like this, you know that they are completely shut down. There is no gene expression going on whatsoever during the M phase part of cell division, because these chromosomes are completely condensed, right? They're looped upon loops upon loops, so that they can be divided more easily, right? And so in mitosis, right, they just go down like so, and they get pulled apart. In meiosis, these chromosomes are actually paired up, you know, the base by base, while they're in this state. And then they condense down together to make those small tetrads that, you know, have undergone recombination. And the reason for that is that if you try and separate up a bowl of spaghetti, essentially you're going to break the spaghetti, right? And so these chromosomes have to be packaged down much, much, much more tightly to be able to accurately separate them during M phase. And then as soon as telophase occurs, right, they decondense because you need to have them in their uncondensed format to be able to access them to express genes, right? And so that's also one reason why we have interphase, is so that we can do all of the gene expression we need to create the things that we need to divide a cell before you shut down your chromosomes and they're completely turned off. So this heterochromatin versus euchromatin can be per chromosome, right? Depending on whether it's in interphase, stationary phase, or M phase, right? So it's kind of decondensed or supercondensed, right? And it's all heterochromatin. It can be by region of a chromosome, right? So the ends in the middle are generally condensed and inaccessible, right? So they're not expressed versus the arms. And also it can vary just depending on context, right? So even the arms shuttle between chromatin and heterochromatin depending on need, right? If you're not transcribing a gene from that region, you shut it down because it saves space. Then when you need to access that region, you open it up, transcribe the genes, and shut it down afterwards, right? So it's a really, really efficient way of making the most of the space that's available, yet still being able to do what you need to do as a cell. Okay, so that's pretty much brought us to the end of today's stuff. And on Wednesday, we will dive into transposons, which a large part, not all of it, but a very large part of our genome is made up of transposons. And actually, the more you read about and the more we study transposons, the more we realize that they're actually really important for our own evolution, which is kind of cool as well. But we'll start on that on Wednesday. In the meantime, have fun, everybody, and I'll see you then. Bye-bye.